let's get started. Because yeah, okay, let's get started. And people on Zoom can hear me. Perfect. I see one thumbs up. I'll assume the rest are all fine. Great, 2 thumbs up. Fantastic. Okay, let's get started. So welcome to Stats Crash Course Session 4. Today's topics are on repeated measures ANOVA, fixed and random effects, and linear mixed models. I will say people have written textbooks on this particular topic, so consider this a very brief introduction to it. Today's session is actually reasonably short, but that will mean we'll have much more time to interact if you wanted to ask questions about it. I really cannot profess to be any form of expert on this. As I said, people can literally write textbooks on this topic. But yeah, linear mixed. So what we've taught about in the previous sessions were mostly about main effects ANOVA, one-way, two-way ANOVAs, and all of these. When we talk about one-way ANOVA, two-way ANOVA, these are all fixed effects. These are all things that we call them fixed effects. We don't actually let them, we don't allow them to have random effects. So now, to explain the background of this, you can imagine, so one-way ANOVA, two-way ANOVA, these sort of things came about probably in the second half of the 20th century, and there were things that you could actually do by hand, and you could easily calculate them. And so that's why people did them. And then repeated measures ANOVA came in the latter half of the 20th, latter half of the 20th century, because they're also reasonably easy to calculate by hand. Now, fixed and random effects and linear mixed models now supersede a lot of these because they're more computationally expensive, and they are not always possible to do by hand, and they're actually computationally expensive the more complex the model is. So I'll just keep that in the back of your mind as we do this, because this does need to be done on a computer, just on a regular desktop. It's totally fine. That's on your laptops. We can run linear mixed models very easily. But that's just sort of the thing. Like, linear mixed models have come into fashion now in the 21st century because of the ease of computational power, and yeah. But I will go through repeated measures ANOVA because that is a nice segue, sort of. It's not really an intermediate between linear mixed models and one-way, two-way ANOVAs and the such like, but it's a nice other version that people used to do. So if you have some of the older supervisors, they'll be like, whoa, why don't you just do repeated measures ANOVA? Even though we have better tools now, specifically linear mixed models, but they still might. And I'll explain the pros and cons as well for using repeated measures ANOVA. Okay, so repeated measures ANOVA. Now, sometimes they're also called within subjects ANOVA, depending on what textbook you read. So let's using the BLAG data from the last exercise, and so it's just from exercise 3, these 40 BLAGs. You recruit 40 BLAGs, fictional alien species. You assign randomly each BLAG to receive drug X at 5 different doses, and at 0, 1, 5, 10, and 20 milligrams per kilogram, where 0 milligrams means the BLAG received a placebo. You then measure the BLAG strength after drug administration, with strength measured in arbitrary units from 0 to 100. The order by which you collect BLAG strength after dose administration is according to a balanced Latin square design, which again we talked about last session. You also collect height in centimeters and sex data for each BLAG. Now, for repeated measures ANOVA, your input, you have a dependent variable. In this case, it's strength. It's a continuous variable. You assume it's normally distributed, and instead of assuming homoscedasticity, you assume something called sphericity. Now, what is sphericity? It's basically like homoscedasticity. It's just basically it's equality of variance of the differences between levels of this within subjects factor. So essentially, if you minus the mean from each subject, you assume the variation is the same, but for every different condition. And how do you test for sphericity? You can use Mauchly's test. So if Mauchly's test is violated, you can use Greenhouse-Geisser or Hunefeld correction. Why do I include all these random things? If you use SPSS, it does that for you. It has like a Greenhouse-Geisser. Like if you just punch in, let's do a repeated measures ANOVA, it'll immediately automatically help you also calculate the Greenhouse-Geisser and Hunefeld correction alongside your repeated measures results. Now, with regards to within subject variables, these are things that vary within subjects. So if you imagine, we've subjected one subject to 0, 1, 5, 10, 20 in a randomized order. So each person's been measured, what, 5 times. If they've been measured 5 times, they have a within subject variation, and that's the strength after each different dose. However, they also have a between subject variation, things that vary between subjects. So for example, if strength as the dependent variable, then what's within subject variable? Well, strength against dose, but what's a between subject? What about sex? Sex is different between subjects, so that would be a between subject. And I hope that comes quite intuitively to you, because that would be, yeah, these are things that you have to intuitively kind of just be able to understand based on your experimental design. So if I ran a repeated measures ANOVA on the data that I presented last week, on the BLAG data, you could get an ANOVA table that looks like this. And so in here, you have the error ID, error within. Now, again, R likes to call it error. Some other people prefer calling it residuals. So in the error ID, you can see that this is actually between subject variability. Between subject variability. And this is within subject variability. Conveniently, it says error within. So this model recognizes that dose is a within subject variable because it's repeated across, whereas you've specified sex and height to be between subject variables, because sex differs between people and height differs between people. And similarly, as you might recall from last week, if you have statistically significant main differences, if you will, if you have differences, then only if you have significant differences, then you can conduct post hoc tests, in which case you can see how they have their 5 different doses, 0, 1, 5, 10, 20, and then I can compare between 0, 1, 0, 5, 0, 10, and so on. And you can also do a p-value adjustment, and you can see I used the Tukey method of adjustment. So that's all possible, and if you're wondering how to do that, well, that'll be in the exercises. So how would you report a repeated measures ANOVA? Well, in the method, you'd say a repeated measures analysis of variance was conducted in R version 4.1.0 using the base stats library. Post hoc tests were conducted using R library EM means version 1.6.1 or whatever your version number you use. Then your results could be BLAGs exhibited significant changes in strength in response to drug X times dose administration, and then again report your F value and your degrees of freedom and P value. So that's from the ANOVA tables. You should report whatever you got from these values. But then using post hoc tests, using Tukey adjusted post hoc analysis, differences were observed between placebo and all groups. And if you're specifically interested in one subgroup versus one subgroup, then you can just add these in, 1 milligram versus 10 milligrams. And then as is here, the estimate is often called beta. Beta equals whatever. And then T equals 4.89, p-value is whatever. So I believe I specifically highlighted the ones that were statistically significant. Some of them aren't, but yeah. And I think that's all I had on repeated measures ANOVA. Let me just quickly check if I talk about the pros and cons. Right, I do. Okay, so in the linear mixed model, now moving on to linear mixed models. Now they're sometimes also called multi-level modeling or hierarchical linear models or mixed effect models. So first of all, you might be wondering, well, why do we bother using linear mixed models? Well, repeated measures ANOVA does not handle missing data very well. If you had, so for example, if one blarg just for example, missed their appointment and couldn't make the 20 milligram per kilogram, you'd have to delete all of their data completely. So if you have missing data, don't use repeated measures ANOVA. What you could do if you had that is you could like you could put in a value that you think is probable. Yes, people do that. But it's just a bit contentious how you do it. A lot of people just do, it's just take the average of the before and after or whatever value. There are multiple ways, and there's other things like multiple imputation that can be done, and that involves some complex chained equation stuff. And it's pretty easy to do in R, but doing that's always a little bit weird. So linear mixed models, however, can handle missing data much more capably than repeated measures ANOVA. Repeated measures ANOVA does not handle further hierarchical data very well. So for example, in each blarg, well, I think we have 40 blargs. So what if the blargs were in families? Some of them had families of 5, some of them had families of 8, or some of them had families of 6. What about there are different families, and different families had similar strengths and different strengths? And then what about they're from different tribes? So 3 families are in one tribe, 2 families are in another tribe. And what if there are tribe effects, where maybe one tribe is hunter-gatherers, the other ones are I don't know, farmers or something, and so there were tribe effects. And then what if you nest them again further? I mean, maybe one of them is a different subspecies or whatever. When you have a lot of this hierarchical data, linear mixed models will be much more capable about handling that. Repeated measures ANOVA does not handle that very well. Okay, and also linear mixed. So when you do repeated measures ANOVA, it's always a bit weird. And look, there's always this assumption of independence, right? However, I mean, when you measure a blarg 5 times, it's not really independent data, because the data does depend on another. So let's say blarg number 1 elicited with the 0 a strength of 50, and then after you gave them a dose of 1 milligram per kilogram, they might elicit a strength of 51. And then after you gave them a dose of 5 milligrams per kilogram, they might elicit a strength of 55. But the thing is, these values are all, those 3 data points, 50, 51, and 55, they aren't independent from each other. Why are they not? Because it's the same blarg, right? If you do it truly independent, if it's truly independence, then it would just be a random number. But in biology, because you're often measuring multiple values per person or per subject or per animal, or per cellular array, whatever, it's often not independent. And unbalanced designs, if you have, for example, 40 blargs, if you had like 30 people versus 10 people, that is an unbalanced design, and repeated measures ANOVA doesn't handle that very well either. And often when you design an experiment, you're probably more likely to put more subjects to undergo the treatment group rather than the placebo group, for whatever reason. Well, actually, maybe not. Maybe you're more inclined to put them into placebo group. But some people, I mean, I know in animal physiologists, they may just put most of them under the treatment group, because they just want to know what happens when you actually do something to them. Why would you waste an animal which costs hundreds of dollars into the placebo group where you supposedly do nothing, right? So often when you have this imbalanced design of overweighting one group, for example, more people into the treatment group versus the placebo group, that's repeated measures ANOVA doesn't handle it too well. If your N gets too small, for example, an N of 4 versus N of 9, that's getting pretty small. So yeah. And also, why use linear mixed models? Well, the strength of it is because you're literally using mixed models, and the mixed refers to the fact that you have random effects and fixed effects. Now, random effects are it's very difficult to explain what random effects are. I will explain it later down here. But yeah, that's why they're called mixed effects. because they incorporate both random and fixed effects. If it only incorporated fixed effects and you had no random effects, then what you're doing is exactly the same as what I've been explaining: one way, two way, and over, and etc. So yeah, if you had, and when I say one way, two way, I just mean one independent variable, two independent variable, three independent variable, four independent. You could have 10 way ANOVA, which means 10 independent variables, but generally speaking, that's probably not a good idea. And you can also incorporate Simpson's paradox, which I will explain below. So first of all, what is the, okay, and also your input for a linear mixed model, your dependent variable is typically a continuous variable. You typically do not use an independent variable. Sorry, for your dependent variable, it's typically going to be continuous. It won't be discrete. And we'll cover that next week. Okay, let's move on to random versus fixed. And if there are any questions, just yell out. So a random variable. A random variable is assumed to be measured with measurement error, and the values are intended to generalize to a much larger population of possible values with a certain probability distribution. Okay. No idea what I just read, but I wrote this apparently last year, so no, I do. I think I do. Okay, so basically the idea with the random variable is that you're collecting from a variable that you're not necessarily interested in, and so you're not actually going to put parameters onto that kind of, and by not putting parameters onto that, you allow it to vary a little bit, because you acknowledge, for example, everyone here, we'll all have different heights, and we know that biologically we're slightly all different, so we don't really care about this particular aspect, so we allow it to vary a little bit. Whereas for fixed variables, we assume we're collecting it without measurement error and that the values generalize to a population, and that will be relevant later. So again, the differentiation between random and fixed effect is often dependent on experimental design and your research question. Okay, let's talk about Simpson's paradox. What is Simpson's paradox? Simpson's paradox is that the trend disappears. Sorry. The trend appears in aggregated data but disappears or even reverses when groups are combined. Simpson's paradox: the trend appears in aggregated data, but disappears or even reverses when groups are combined. Now, what does that mean? So, for example, oh, right, you couldn't even read that. That's better. So, for example, in our blarg example, let's say strength versus dose. Let's just say we saw something that looked like this. So we saw a trend that looks like this. So strength is negatively correlated with dose. That's what the data would seem to imply. However, Simpson's paradox is when you actually subdivide it into groups, what if you had 3 different groups in this data, and instead the data looked like this, and then like this. So the trend is now slightly different, and this is what Simpson's paradox is trying to get at, that even though you have an overall negative trend when you have when you sort of aggregate data, when you put them into their groups, they might have an opposite effect, or the effect just disappears. It doesn't need to reverse. But yeah, the idea that we see a positive trend when you put them into their own groups. And so that's also a strength of linear mixed models. You can actually identify these trends that are occurring. So yeah, okay. So model diagnostics and assumptions, right. So the residuals for the entire model should be normally distributed and have equal variance. So I think I alluded to this, I think in session 1 or session 2, but rather it's the residuals of the model. And do I actually ask you to do that? Okay, I can show you how to actually do that later, but in most statistical analysis, usually it actually is asking that the residuals of the entire model should be normally distributed and have equal variance, rather than the data should be normally distributed. Often the case is that the data being normally distributed means that the residuals are normally distributed, but that's another story. Okay, but I wanted to draw something else in this space. Now, let's just draw, so in this example, I'm going to use the model strength is dependent on dose and ID. So dose has 5 doses, whereas ID has person number 1, which I'll write in blue, person number 2, which I'll write in red, and then person number 3, which I'll write in black. Doesn't show up quite well. Well, it shows up all right. So over here, I'm just going to draw this into quadrants. So let's just start off in this quadrant. So we're going to do a fixed intercept model. Fixed intercept, but a random slope model. Okay, I'll in this quadrant, we'll write fixed intercept and a fixed slope. In this quadrant, we'll write random intercept plus a fixed slope. And in this quadrant, we'll write random intercept and random slope. So we're going to start in the top right quadrant where we have a fixed intercept and a random slope. Now in this data, we'll have, so I'm going to just try and draw some data here. And I'm going to draw the same data in all 3. So in this data, we'll have 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. And also 1, 2, 3, 4, 5. So I'm just going to draw the same data quickly into all of these. Strength versus dose. Strength versus dose. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 3, 4, 5. 1, 2, 4, 5. Okay, so what I've tried to do is replicate the same 3 graphs, just replicate the graphs 3 times. Oh, sorry, team. Okay. Okay, now in this case, in the top right, I'll just wait maybe another 20 seconds. All right, so in the top right, we're going to assume a fixed intercept, but a random slope. So when we say intercept, we literally mean the y-intercept. So we assume everything will start at a fixed intercept. And so for the blue person, for ID number 1, the blue person will have a fixed intercept, but the slope is allowed to be random. So a random effect slope. So we can allow that. So based on this, you might get a slope of here, and you might, for the black line, you might get something fixed intercept. So same intercept, fixed intercept. And then, gosh, if you try to draw a straight line for the red one, that'll just be slightly weird. So it might just, that's not exact, but it'd be similar to the black one, maybe. So that's a fixed intercept and a random slope. Does that really describe the data well? Probably not. And that's why you, in this case, you probably wouldn't use it. Now, what about the random intercept and fixed slope for person number 1? So if it's a random intercept, you allow it to cross the y-axis at a random place. You allow it to vary around here, and you also, but however, the slope must be the same. So it would look like this. It would probably look like this. And then the red one will also have to have the same slope. So we have the exact same gradient. So the 3 colored lines have the exact same gradient. They have the same, they are parallel lines, but they just have a random intercept. All right. So that's when you allow a random intercept, but you have a fixed slope. Does that describe the data reasonably well? I think it does. Now, what about the last one then? Random intercept and random slope. So you acknowledge that the intercept for every individual will be slightly different, and you acknowledge that the slope will be slightly different. What does that mean then? It just means that you can't really generalize that, oh, people will have, so in this case, sorry, when you have a fixed slope, you can be like, oh, generally there is a relationship between strength and dose because we're fixing it. We're fixing it to be a gradient of, I don't know what that gradient is, let's just say it's a gradient of 5. We're saying that we're generalizing to the population that the gradient is a fixed slope of 5. So that way we can report it in our data saying, yeah, it's 5. But we acknowledge that random intercept, people have a random intercept, so we won't be able to report that data. So what about random intercept and random slope? What does that look like? Well, we allow it to vary on the y-axis, the intercept, and we also allow it to have the random slope. Let's just draw it for the blue line. So it'll probably be ID person number 1, looks like that. For the black line, it'll probably look like that. And then for the red line, or it might just be a horizontal line-ish. So you're allowing every person to have their own intercept and have their own slope. Now, the thing is, just keep in mind that this is when we have a continuous axis, right? A continuous independent variable. So what's the dependent variable here? It's strength. And the strength variable is certainly is a continuous variable. And what's the independent variable here? Well, the independent variable is dose. And now we're saying every individual will have different responses from the relationship of strength and dose will be defined in a different way. And so we're assuming that dose is a continuous variable. That's why I'm drawing lines. If we're talking about discrete variables, if you're going to discretize every dose, then you won't be able to have slope variable, right? Because slope assumes dose is a continuous variable. So if you were to discretize dose, you would not be able to use random slope. These would not be able to exist. You just use this one. But yeah, if you do model with continuous variables, it's often useful to have a slope. So it's just getting used, understanding sort of like when you allow things to be random effects or fixed effects, that's sort of what you mean. You're allowing them to have random y-intercepts, and y-intercepts, allowing them to have random effects. So if we're to say, let's just ignore the slopes. If you just say a random intercept or a fixed intercept, let's just say you allowed sex for whatever reason to be a random effect. You would allow every person to either be, if it's male or female, you'd allow it to be low or high, 0 and 1, that'd just be that'd be a fixed intercept, sorry. But if you allowed it to be random, you'd allow every different sex, i.e. 0 and 1, which is just male and female, to be 2 different effects, which now random effects you should only really allow random effects if you have more than like 5 levels of, or 10, like this, when you have so many different levels. For example, that's why ID is a perfect example of a random effect, because typically you might have 10 people and therefore you're allowing 10 people to have different parameters, slightly different parameters. But if you put sex as a random variable, but you only really have 2 samples of sex, male and female, you might sometimes have a third sex, but that's not enough to really allow it to be randomly allocated. So you usually only should allow something to be a random effect when it's got more than like 5 to 10 or even more than that. Because that, if you imagine, you're trying to group all of these onto like a bell curve, essentially, like a normal distribution. So you should only allow it to vary like that. Yeah, right. Mixed effect models allow for random effects, whereas groups or clusters, repeated measures from a participant, are allowed to have slightly different parameters. I don't remember what I was on when I wrote that. Yeah, okay. And yeah, now is probably the time to move on to the exercises. Do I have 1 page of exercises or 2 pages of exercises? Just 1. Great. Okay, well, let's move on to the exercises then. Okay. Okie dokie. Now what? Now I need to share screen from my laptop. Let's use that virtual computer. Okay. Today we're on session 4. You still can't see me yet. Give me a second. Okay, so today we're on session 4 now. Okay. That should go away. Okay. All right, so, okay, I don't know why that reads session 3, but that shouldn't read session 3. This is session 4, I think. So I'm just going to run all the library things as per usual. And my computer's slow because it's also running Zoom at the same time. Now you want to get your working directory. So for those who use a Mac, you can always, I think this is the Mac OS one. So like, I think it's X. So yeah, so for me, I'm going to go get my directory, and I'm going to set my working directory to be that, except R only likes using forward slashes. So I'm going to change them all to forward slashes. So that's my directory, but you're going to want to set your own working directory to whatever it is. Then I'm going to read in data frame 1, in which case it is called seminar4_df1_blarg.csv. Is that correct? Blarg.csv. Yeah, it is. Okay, so load in the data set. So this is the same example that I talked about in the thing just then. Great. So df1, and then if we just view the structure, always do str df1. That's just the useful command to do. It views the structure. So you can see we have ID factor, ID variable, which is int stands for integer. So let's just view df. No. Let's just view the head. Let's just view the head of df1. No, let's view df1. So if you view df1, it looks like this, 1, 1, 1, 1, 1, and then you can see dose 0, 1, 5, 10, 20, strength was all of this. And you can see the height, what's the same, sex, what's the same, and order recall that we had different orders for these people and for these blogs. So remember that we want ID to be a factor variable, not a numerical variable, because you can't have person 1.5 and person 1 and 2 aren't really related. Well, not necessarily related. So so we want to change this into a factor variable. So that's what this line does. We also want to change dose into a factor variable. So in my field of research, I'm used to changing dose into a factor or a discrete variable. Some statistical purists may view that not acceptable and consider that more as a continuous variable. But I'm running the course, so and then sex we'll call a factor variable as well, and order we'll also definitely call a factor variable. Now we can re-look at the structure. You can see I've changed it into a factor variable, numerical, factor. You can view the head of the data, and you can also view df1 again, which I don't want to view, but cool. I think in the first question it says conduct a 5-way ANOVA. Strength is dependent on dose, on ID, on sex, on height, and on order. Now that's a 5-way main effects ANOVA, so that's not a linear mixed effect. That's not a repeated measures ANOVA. That's, if anything, from last week, but it's just to connect the concepts. That's how you do it. It's very simple. Model 1 is equals LM. LM is linear model. Strength is dependent on dose plus ID plus sex plus height plus order, and then comma, you can always do data equals df1. So if you just go question mark LM, you can also look at how do you define this, and you can look and then the help will pop up. It expects the first argument to be a formula, and you can see this is considered a formula because it's got a tilde symbol. Strength is dependent on dose plus ID plus sex plus height plus order. That is a formula. The second argument it expects is df1. So I could actually just do, now this is just the random programming. I really hesitate to call this programming, but anyway, I could say data equals df1, formula equals strength is dependent on dose plus ID plus sex plus height plus order. I could write that and it'll have the same effect because I've explicitly said data equals, and that should give me the exact same thing. It does. Okay, I thought it might error out, but it didn't because I explicitly said formula equals. However, if I don't say formula equals or data equals, then it will assume that the very first argument is whatever is in line with the function. So just be careful, just be mindful of the what the functions expect. And the function LM is stands for linear model. You can view more by doing question mark LM, which just opens the tab we have on the right. Anyway, now that we've done this LM, I'm just going to run the original one, and then we can run an ANOVA on model 1, and it gives me that. So we can see that it is statistically dose, ID, and order is statistically dose and ID are statistically significant. Order is not. Do we want order to be statistically significant? Generally speaking, we hope not, because that would be, if it's not statistically significant, that's good, because that would mean if it was statistically significant, it would assume that if you gave person drug 20 milligram per kilogram after 5, it might change the way it impacts the data in the future. We don't generally want there to be order effects. However, it's just like, do you want there to be a washout effect? Generally speaking, you want when you have a crossover design, or when you repeatedly sample someone, ideally you're sampling independent additive things. When you start dealing with interaction effects, as we talked about last time, it gets even more complicated. Bends your brains in ways you didn't even know they could bend. So yeah, but you might also wonder, but wait, where's the height variable? Where are all the other variables? What about sex and height? And that's a great question. I believe it's because of it's just because of the way R is programmed. It hasn't the degrees of not the degrees of freedom, the sum of squares that are calculated by the sex, and height is actually already incorporated in these differences. So if you imagine, if you put strength is dependent on sex, for example, if you put strength is dependent on sex, well, let's just run that quickly. So this is just, this is a tangent, so ignore me. Temp model equals linear model strength is dependent on sex, df1. If we did this, and then n over temp model, imagine now if we did that, so it means that we got all the males into one group and then all the females into the other group, then we measured their sum of squared deviations from the mean, and then we did that. But the thing is, what if we did this? What if we did temp model 2 equals linear model strength is dependent on ID and df1 and over temp model 2. What if we did this? You can see how you can see how ID encompasses 16,993 sum of squares, whereas sex encompasses 10,844. What I'm trying to say is that the variation described by ID actually incorporates sex, because within the different people encompassed by the 16,993, that includes all 40 different people. It includes the variation between all 40 different people. But the thing is, that variation between all 40 different people is actually also incorporated by the fact that people have different sexes. Some of them are male, some of them are female. So that's why when I told R to run model 1 back here, let's just delete this. When I told R to run model 1, it was like, okay, I ran dose sequentially. I ran dose because I identified dose was the first one. Then I ran ID. But then when I told it to run sex, it was like, no, but ID, when you measure ID as a fixed effect, it actually already encompasses the sex variable. Now, I'm not sure if that made much sense, but I hope it did. Okay, all right, let's talk about repeated measures ANOVA. I think question 2, conduct an equivalent repeated measures ANOVA. So let's do that, RM ANOVA, repeated measures ANOVA, and then you can do this, summary, RM ANOVA. Now this time that was so you can see within subjects, sorry, ID, this is between subjects. So you can see between subjects, you now have a sex variable, you now have a height variable, and then within subjects, you now have all of these variables. And dose and order are definitely within subject variables because you've repeatedly sampled every single person 5 times, and that's why there are 4 degrees of freedom, and you also gave them different orders, and that's why. Now I'll just bring your attention to 10,844 is also the same value, I believe, here when I just did strength is dependent on sex, and again that's 10,844, and so a lot of these numbers are repeating. However, just recall when I talked about how do you calculate the F ratio, it's always the sum of squared, sorry, the mean square. So in this case, mean square is just sum of square divided by degree of freedom. So it's 10,844 divided by the residuals. So actually, when you're talking about between subject variation, essentially you're getting, so I should probably explain this command first. So if you just read this command, RM ANOVA 1 equals AOV, strength is the dependent variable, is dependent on dose, sex, height, and order, plus, and then when I do error, open bracket, close bracket, ID, I'm saying that ID is the within subject error. This is just the way R likes to notate it. Now, by identifying this as the within subject error, it's now understood that, okay, so I'm going to sequester all the sum of squares from this ID variable into a bucket. And what bucket have we sequestered it into? Let's just have a look. We've sequestered it into this. And this is all the between subject variable. If you add 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 5511, you'll note that it adds up to 10,844 plus 638 plus 55 16,993, because that's all the possible variation we have between subjects. All right, so you might have recalled that I called SS, sum of squares, I called that as like the units of the ANOVA. That's how I view it, if that's of any help. Not sure if it is, but hopefully it is. And then within subjects, you have dose, order, and residuals. So yeah, so I mean, let's just calculate. Okay, sorry, df1 strength, var df1 strength, just calculating the variance of strength times, I think I have to variance, and then I have to think I have to times 40. No, I'm getting confused. I think it's times 200. So they're 18,000. That's the total sum of squares, because I'm just multiplying the variance times by n. So instead of getting mean square, which is essentially variance, I'm multiplying it by 200 because there are 40 blogs and we record them 5 times each. So I'm just multiplying it out. There are 18,000 sum of squares to divvy up. And I think 15 or 16, whatever I just said, 15, 16,000 of it was gone to between subjects, and the remaining 4,000 sum of squares is within subjects. So that's how I sort of view repeated measures ANOVA. And then you might be asked to perform the post hoc tests for the repeated measures ANOVA. 4.1.3, library. You need to open up the library, not open up, sorry. You need to call the library EM means, and if you haven't installed it, then install library EM means. You can do install.pack, sorry, not comma packages, but install.packages EM means. That's what you can run in order to install that package. But here, I've already installed it, so I'm just gonna cancel that. If you wanted to know what version, you can run this line. So if I run help package equals EM means, you can see it's version 1.6.1. So that's why in my reporting, in my journal article, I'll write I used EM means version 1.6.1. And then how do you actually do, so we have summary RM ANOVA 1. We have this. Now, how do we run the post hoc tests? Well, you can simply do that. So you can add that to make sure this is the same as that, and you can run that, and then you get all of these things. So you get all of these outputs. So first of all, you can look at the different contrasts, because you have 5 different groups, right? 0, 1, 5, 10, 20, and then you can do what? 0 against 1, 0 against 5, 0 against 20, 0 against 10, whatever, and 1 against 5. So there are many comparisons you can do, and then you can see which ones are significant. And then, yeah. Results are averaged over levels of sex and order, which is fine. We do want that to happen. And then just be careful. Some predictors are correlated with the intercept. Results may be very biased. Yes. Cool. Okay. Okay, now do the same thing, but okay. Now you can perform post hoc tests. Oh, we did that. Now you can do the same thing with linear mixed models. So when you use linear mixed models, I use LMER. So let's just have a look at what LMER is. LMER is fitting a linear mixed effect model with REML, or maximum likelihood, and it's from the library LME4. LME4 at the beginning, I already called it library LME4. But yeah, that's a very popular package to use. In fact, it's kind of like the standard package to use. So strength is dependent on dose, sex, height, order, and in order to call something as a random effect, so now we've made ID or person a random effect. Each blag is now a random effect. And I believe this is like 1 is meaning fixed intercept, and then 0 is, I think, random intercept. It's slightly forgotten. You can always Google it very quickly and it'll tell you. But anyway, so let's do that. And then we can get that. Now that's not very informative, so let's do summary. That's not very informative, so let's do ANOVA. And that's more informative because now we have this. So you can see the total intercept is statistically significantly different from 0. That's not that useful, really. Dose, now remember how I said you need to have a significant sort of main F significant before you can do post hoc testing. So we're probably most interested in dose. So dose is definitely significant. You can see 132.56 divided by 4 equals a very small number, and then get the thing, change it into p-value, and then it's very significant. Then there are significant sex effects, and there are significant height effects, but there's no significant order effect. Great. So that's what you can do in a highly controlled experiment. Okay. Great. All right, let's move on to the second question. A between group study, rats. Okay, so let's read in the rats data. Let's have a look at, I'm going to change ID and sex and drug all into factor variables. Why do I have 2? Oh, okay. Let's read the questions then. You have 24 rats. You have 12 males and 12 females. You record their weight. You randomize the rats to receive control, n equals 6, vehicle, n equals 6, or drug X, n equals 12. You performed a behavioral test recording at baseline, on experiment day, 1 week post-experiment, and 2 weeks post-experiment. You are given the same data in 2 formats, wide and long. So question 1 is, how many individual recordings do you have? Okay, let's just run all of this. Let's have a look. Let's view. How do I view DF2A? Okay, so how many individual recordings do we have? How many rats do we have? We have 24 rats, and then we're recording them 12 times 4. I think that's just it, right? 12 times 4, 24 times 4. 24 times 4, I believe, is 96. Okay, let's just view the long format, which will, if I view the long format, you can see how ID rat number 1 was recorded at baseline, experiment day, week 1, week 2. And then you can just scroll all the way down, and there are 96 recordings. Awesome. So that answers that question. How many individual recordings do you have? We have 96 recordings. So how many degrees of freedom do we have? Roughly 95. But just remember that when you resample one person, so like in this case, each rat has been sampled 4 times. When you resample everything, like that's kind of pseudo replication. It's not fair to say, because we have 96 recordings, therefore its degrees of freedom is 95. But like, there's something inherently wrong with that, because you've recorded each person 4 times. Doesn't mean that's, it's not exactly right. That's why when you do corrections and such, like corrections for degrees of freedom, they usually correct the degrees of freedom, because there's something slightly fishy about that. Okay, perform a repeated measures ANOVA. Let's cross this out. Perform a repeated measures ANOVA, and I believe that's what you want to do. Let's have a look. So the score is dependent, the behavioral test score was dependent on drug, weight, sex, time, and error ID. That's how you notate which one's the within subject error thing. And now this is the this is between or within? No, this is between subject error. So between subjects, they have different weights, they have different sex, but within subjects, they have time. So it's like before the experiment, on the study day, 1 week post-experiment, and 2 weeks post-experiment. So that's what you can do in linear repeated measures ANOVA, and then you can do the same test on using linear mixed models, like that, LMER2. Okay, let's just do that. And I'm just gonna run both of these. So what does that look like? We've got repeated measures ANOVA, and we have the LMER, and you can see how some of these values look very similar. So okay, yeah, interesting. But yeah, but that's how you would run a linear mixed effect model and linear mixed effect, or linear mixed model and a repeated measure ANOVA. And now generate a graph for publication, which highlights the effects of, and then I stop typing. What? Okay, I promise this will be fixed for next year. Okay, well, let's just say it highlights the effect of drug, right? Because that would be, and I don't have any code for it. Well, let's just make it from scratch since we can. Okay, so let's call it rats graph equals. So first of all, in order to make a ggplot, you need to initiate it by calling ggplot. So first of all, you need to have library ggplot, and it's actually this ggplot2 library, which I've already called at the beginning, which you can see over here. I've already called ggplot2, so that's, I don't actually need to call it again. So let's just delete that. Ggplot. What data do we want to use? I will tell you R prefers to use the long format. So I'm just going to question mark ggplot. Ggplot is the command. The data is the first argument. So I'm going to do the first argument, df2b. And then now the mapping. And mapping is an AES. AES stands for, doesn't stand for, it's short for aesthetics, where you have to do the x-axis, the y-axis, and the such like. So let's just have a look. What would make sense for this data? We clearly want to know the effect of control vehicle drug. So we clearly want to know the effect of condition, but we also want to know the effect of time. So in your mind, what do you think makes sense? What should be the, okay, well, first of all, what's the y-axis? What's the dependent variable? Almost certainly it'll be the effects of behavior, or the behavioral test, right? So what did we call the effects of behavior? So let's go ahead. df2b, we called it score. So the y-axis will be the y equals score. X-axis, now let's just call it time, because x-axis is a good time one anyway. So x equals time, and it's time, all lowercase, time. And then we also want to do drug. How can we denote drug? What's another way we could include it? And because we had control vehicle and drug X, what can we use to put 3 dimensions onto the plot? We can use color maybe. So let's just do color equals drug, but I'm also going to call group equals drug. And now I'm going to add. So first of all, if you just ran this, and now let's make rats graph, nothing would plot. It would just plot something blank because I haven't added any geomes. So let's add some geomes. So okay, I have, okay, that's okay. Okay, so let's add geome point. Let's see if that does anything fun. Cool. So you can see how I've actually plotted all the values on here. Now I don't actually want to plot all the values. I just want to plot like means and standard errors of the mean. So in order to do that, I can never ever remember the code which does the summary. So what I'm going to do is I'm going to open my previous seminar's code and then copy paste that code and copy paste some other people's code and put it here, because yeah, that's like the whole point, right? So I use the ddply command. So let's do that, ddply. So dfsum, ddply. So okay, df2b sum. So ddply df2b. And I want to summarize, and let's just go view head df2b. So what's going through my mind? I want to summarize this data frame, and I want to summarize it against time and drug, because I want every combination of time and drug, and I want to summarize it. And so now what do I need? I need score mean equals mean score, score SE equals standard error score, So let's run that and let's do that. And so now I have that. So I have time at baseline, I have the control mean and standard error. At baseline, I have control the vehicle mean and vehicle standard error. At time baseline, I have drug X and the score mean and score standard error. So that's what I'm going to do. So let's just change this to df2b sum, and let's see if this works better. So instead of plotting the individual values, let's plot just the mean values. Oh, the y-axis. So what do I have to do to make this plot? What's wrong with this code and why did it give me an error? I'll just show you it. Yep, it doesn't exist. So what do I have to do to change it to make it work? Score mean. Yep, absolutely. Yeah. Awesome. So we just want to plot the mean. And perfect. Rat's graph and the score mean pops out. So now we don't have to plot like a massive chunk of data that's ugly. We now can do that. Now, also, if you want the size of these to be massive, you can also, 10 is going to be really funny. Oh, that's, nature worthy. No. Let's just do 2. 2 is a nice number. Okay, and then maybe just for fun, let's do geom underscore line and size equals 1. Let's just do that. The reason why I put size equals 1 is because I've plotted many graphs using ggplot2 before, and I know that I personally like point size is 2, line size is 1. So yeah, that's what I like to do. You do whatever you like to do. Come on, plot. Oh, did I break something? No, it didn't. Okay. When you do geom line, it just connects the dots up like that. Now that's why I had group. I'm going to remove this group over here. So there's a reason why group is important and color is important. If I didn't have group and then I tried to plot this, I'd be met with an error when I tried to. So each group only consists of one observation. Do you need to adjust it? So when I put line, just know that line and group is important, because when you group things together, it means the line will know to connect which groups together. That's why I included that. So yeah, that just comes with increased experience with plotting. Okay, so anyway, let's keep on going. So what do we want to do? Scale x continuous. Is the x-axis a continuous variable or discrete variable right now? An R. Absolutely, it's a discrete variable, so I've actually typed the wrong thing. Let's change it to discrete. Great, to scale discrete. Okay, and I've already forgotten what the things for discrete are. Oh, right, yeah, name. Name, I'm gonna call it time. Values, so what I want it to be called, let's just call it baseline. Let's have some decorum in here. Experiment day, and then week 1, week, rather than week 1, week 2. Let's do that. Yeah, I like that. Let's plot that again. Each group consists, oh, did I forget to do something? Let's just run that again. Unused arguments, scale, name equals time, scale plus discrete, scale x discrete, x equals time, group equals true, scale discrete x, values of the x-axis. Okay, I'm gonna look at my past code because that usually gives me a clue. Okay, values, maybe it's not supposed to be values. Labels, thank you. Let's try labels. Oh, thank you. Labels. Cool. Oh, and I probably want error bars, right? So geom error bar, and the mapping is AES. X equals time again, but y min equals score mean. The minimum y, do I have to minus the standard error or plus the standard error?
subtract the standard error. And then the y max will be score mean plus score standard error. I think I need one more bracket and one more bracket. And as usual, I might need to define the groups again. That's usually the case. If you don't, oh no, that's fine. The reason why I didn't need to define the groups is because the groups were already, if you don't define the groups, it will just resort to what you initially set. Now these widths look slightly ugly, so I'm going to change the width equals 0.25. I think 0.5 or 0.25 typically works quite well. And then that plots something that looks like this. And that's beautiful, don't you think? Yeah. And then what else do we want? Also, I prefer using theme_classic. So that looks better. What else is wrong with the graph right now? We also want the y-axis to not just have score mean. So let's change scale, y continuous, name equals behavioral. Let's just call it, what's a behavioral test? Let's pretend that we did the tailspin, the forced swim test, seconds. That's a long time to be swimming for a rat, but anyway. And let's do, now the y-axis, I don't want it to only have 2 breaks at 250 and 300. Let's just have a look at the data. You can see that the data will be ranging from roughly 200-ish to about 350-ish. So let's put the limits equals, not 0, sorry, 200 and 350. Now in some journals, they say, no, you need to put the y-axis at 0. You cannot have broken axes, i.e. starting at 200. So if you got to do that, you got to do that. So, but yeah, now also the breaks, let's do breaks sequence between 200 to 350. Let's break it every 10. So let's plot that and let's have a look what that is. That's a lot of breaks. That's too many breaks, actually. Let's break it every 20. But then if you break it every 20, 200 to 350, it won't work. So let's do 200 to 360 and every 20. Maybe that might look nicer. And let's do that. Great, okay. Now I also want to put some asterisks. So how do we do this? So the red one's the control. So how do we want to do this? Maybe, so let's go back to our stats. ANOVA, let's do this one. So when we did the linear mixed effect model, we wanted to basically compare between control. We want to compare everything against control. That's generally a good practice to do. So maybe what we could do is we could do EM means. So let's do the EM means. Okay, EM means. So let's compare between, right. This is comparing between 0 and 1, 0 and 5. Wait, is this even the same exercise? No, this is the wrong exercise. Oops. Okay, so I'm just going to copy this. Where is my EM means thing? So, okay, so where am I? Linear mixed model. Let's do some post hoc tests. So EM means LMER2, I believe. Yeah, LMER2. And now we want to do pairwise between, what would be a natural comparison we'd want to do? We'd probably want to compare this value against this value against that value. So we'd want to compare between, not control and vehicle, drug times time. So let's do that. And it'll take some time because that is complicated. And that gives me a lot of comparisons. Whew. Okay, so the ones we want to compare is control. base versus X base, so that's not significant. Control base versus X base is not significant. Control base versus X experiment. Control base versus X experiment is significant. Well, that's okay, but that's not what I wanted to. I wanted to do vehicle base. I wanted to look at these two. So this red one is not significant between these two, so that's not of interest. Then I also want to compare experiment day. So let's look at control experiment. But you can see how we have to like look through every individual one, and that can be quite time consuming. But yeah, anyway, so let's not add those asterisks and the such like, because that's what you'd have to do. And then you can, if you wanted to add the asterisks, then again, you go back to some of the code that I've given you. And where did I put some? It'll be annotate. Typically I use annotate to do this. Where is annotate? Okay. Typically you'd go annotate, and I think it's text, and then at X equals, now just note that this subconsciously R will think this is X equals 1, this is X equals 2, X equals 3, X equals 4. So at X equals 1, Y equals, let's say 360, we'll call it, I think value. Why don't I just do that? Question mark annotate. And then you can do annotate text label. That's the one. Label. Oh, sorry. Label equals asterisk. Sorry. There we go. So if we wanted to do this, that's what you can do. And I've just done this just to explain. That's how you can add an asterisk to denote that. Anyway, let's move on to the final one. Okay, RCT, a crossover design. You had 6 doctors who recruited a total of 36 patients for you. You have 19 females and 17 males. You recorded their height. The patients will receive either placebo, intervention, or patients will receive intervention, placebo. You randomize them to either receive placebo or the intervention first, 18 in each group. You measure each participant after you give them either placebo or intervention. This means you've measured each participant 4 times. How many individual recordings do you have? So let's read in the data first. So again, I've supplied it to you in wide and long format. So let's view the data, view DF3A. Okay, so we recorded them 4 times. So 36 times 4, what's that? 144. So I think we have 134. So let's just go through DF3B, view DF3B. Yeah, okay, it's 144 values. Typically the long format should have the number of recordings typically, but that's not always true. Okay, so I think I asked you to perform a repeated. The reason why I always ask you to just check how many individual recordings you have is just to keep track of how much data you should be expecting to manage. And so 144 rows, that's usually fine. If you're going to be managing like 10,000 or 100,000, R is going to struggle unless you use Tibbles and Tidyverse, which we won't cover, but yeah, you'll need to learn extra stuff. Okay. Perform a repeated measures ANOVA, simple like that. That's a repeated measures ANOVA, already done, very quick. And then just, yeah, that's the code. Now, I should probably explain, right? So the reason why, okay. So RM ANOVA is score. So what is the score? I didn't actually define, it's just an arbitrary thing. So an arbitrary value we're measuring about these people. We wanted to see the effect of height, the effect of sex, and the effect of order times time, because we're worried about an interaction effect of order. But then we also did error, doctor/ID. What does that mean? So first of all, ID, we're acknowledging the fact that we're acknowledging the fact that every single person will have their own values. So it's so that's a between subject thing. However, what this means is that we're nesting each person under each doctor. I told you I think there are 6 doctors, and some doctors recruited more patients, some recruited less, and we can view that by going to DF3B and can see Dr. F recruited quite a few patients. Dr. A recruited 5, Dr. B recruited 6 to 15, Dr. C recruited some, Dr. D only recruited 1, Dr. E recruited 4, Dr. F recruited another, I don't know, 8 or something, 9. So yeah, so it's just acknowledging the fact that each doctor will have a nesting effect. And why do we do that hierarchy? To account for Simpson's effect, to try and make sure we don't get that Simpson's paradox. All right, so that's what you can do. You can do a summary RM n over 3. Now, linear mixed model, you can, I think I just asked you, perform a linear mixed model. So there we go, that's the linear mixed model, and that's the same sort of way to do it, doctor/ID, and just that's the code, very straightforward. That's not particularly informative. That's not particularly informative, but this is more informative. You can see there, is there an effect of height? No. Is there an effect of sex? Yes. Is there an effect of order? No, thank goodness. Is there an effect of time? Yes. Is there an effect of order against time? Yes. And yeah, that's how you can do. Now, if you're wondering about how to do that nesting stuff, I just Google it. Stack Exchange will always have, not Stack Exchange, Stack Overflow. Stack Overflow will always have some sort of coding help to help you do that. Generate a graph for publication. Yeah, I mean, we could go through and do that, but you can give that a try yourself, and I can help, I don't mind. Yeah. Notice that I just say generate a graph for publication. Often when you are just given your data, your supervisor will just be like, okay, now give me a graph. And you're like, but what graph? Then you have to figure out. So I personally just draw in what I think will be an efficient way to display the data, and then you just got to figure it out that way. So yeah, that's how you do the stats. I hope I've just, the main point of this is to go through the types of statistics that you do. You've got your data, you summarize the data, you do the statistics, and then you plot, and then you add in the frills for publication, and then you save it. That's the point of this, right? It's the point that, so you should be able to do that. And there's a reason why I bothered to go through just then trying to plot that, is because that's what, as postgrad students, that's what we do. Yeah. All right, so I might end it here. So you're welcome to have a try, plot a graph if you want, and I'm happy to just stay around, stick around, and help anyone with that. But yeah, otherwise, thank you for your time. Thanks for showing up again. And yeah, otherwise, if you're heading off, see you in a week's time.